Uh, Good evening and welcome to the ninth live interaction session. Since nobody has joined in yet, I'll start the session, and the video will be recorded, and whoever wants to access the material, they can access it later on as well. So the format remains the same. I'm going to first discuss present and MCQ, and I'll explain the concepts related to that MCQ. So let's get started. So last week we had stopped at question number 15. Uh, we had stopped at question number 15 and we were discussing uh, week 5 questions. So it was towards the end of week 5 questions. Uh, so select the correct statement. Select the correct statement. 1A interneurons always receive excitatory inputs. This is not true because uh, 1A interneurons receive both excitatory as well as inhibitory inputs. They receive excitatory inputs from the 1A afferents and they receive inhibitory inputs from the Renshaw cells. So we had seen that last week. Uh, Let me just go to the previous weeks. So this was the situation. This is the agonist muscle. This is the antagonist muscle. This is the 1A afferent which is sensitive to stretch. So when the agonist stretches, the 1A afferent start firing, and this in turn, the 1A afferents in turn excite the 1A interneurons, and once the 1A interneuron uh, gets excited, it inhibits the alpha motor neuronal pool of the antagonist. So this stretch can be caused due to contraction of the antagonist. So this, because it is now receiving inhibitory inputs from the 1A interneurons, uh, it'll uh, it'll reduce its contraction, and hence this stretch gets reduced. So this is a form of negative feedback through the antagonist muscle. So here we can see that the 1A interneuron receives excitatory inputs. The other situation is we have the agonist here. So alpha motor neuron fires to contract the agonist. The alpha motor neuron also excites a Renshaw cell. So this is the Renshaw cell, and the Renshaw cell inhibits the alpha motor neuron that excited it. So this is another form of negative feedback. So to decrease the contraction, and what it also does is it inhibits the 1A interneuron, and the 1A interneuron, uh, whose action is to inhibit the alpha motor neuronal pool of the antagonist. So that property of the 1A interneuron decreases. So because the 1A interneuron itself is being inhibited, its inhibitory effects on the antagonist motor neuronal pool uh, decreases. So what happens here is, since now the antagonist muscle is receiving less inhibitory inputs, the probability that it will contract increases. So now if the probability that it will contract increases, then what happens is that is acting. So this is called disinhibition. What it does is, if it contracts, it will lead to an expansion of the agonist, right? And so that is another form of negative feedback. So alpha motor neuron contracts, causes contraction in the fires and causes contraction in the agonist, and through this mechanism there's a negative feedback where the action, where the antagonist contracts, which is opposing the action of the agonist. So it will lead to an stretching of the agonist instead of a contraction. So this is one form of negative feedback. So this is what we saw last time. So the 1A interneurons always receive excitatory inputs. That is false. Has somebody joined in? No. Stay in the call. So the 1A interneurons always cause inhibition to the next neuron. Yes, they always cause inhibition. This is true. We can see that here. So the 1A interneuron causes inhibition in the alpha motor neuron. It doesn't cause an excitation. That's a property of the 1A interneuron. Similarly, the 1A afferents always cause an excitation. They do not cause an inhibition. The 1A interneuron receives excitatory inputs from the Renshaw cell, whereas they receive inhibitory inputs from the 1A afferents. So this is wrong. We just saw why this is the case. The 1A afferents actually give excitatory inputs, and the Renshaw cell gives inhibitory inputs. So it's the opposite that is given here. And the 1A interneurons receive inhibitory inputs from the Renshaw cells. This is correct. Whereas they receive excitatory inputs from the 1A afferents. Yes, this is true. And then we have 1A interneurons always receive inhibitory inputs. No, this is not true. We have again seen it why that is the case. Renshaw, the 1A afferent itself excites 1A interneurons. Renshaw cell causes inhibition of the alpha motor neuronal pool of the antagonist muscle by inhibiting the 1A interneuron. Okay, let us read this again. Renshaw cell causes inhibition of the alpha motor neuronal pool of the antagonist uh, causes inhibition of the alpha motor neuronal pool of the antagonist muscle by inhibiting the 1A interneuron. No, actually, it inhibits the 1A interneuron, which since the 1A interneuron is inhibited, its inhibitory effect on the alpha motor neuronal pool of the antagonist decreases. So, in that sense, the action of the Renshaw cell causes an excitation in the antagonist muscle, not an inhibition. The 1A interneuron always causes excitation in the next neuron. No, it causes an inhibition. And the Renshaw cell causes disinhibition of the alpha motor neuronal pool of the antagonist muscle by inhibiting the 1A interneuron. Yes, this is correct. So we have seen this. It causes a disinhibition. So it inhibits the inhibition of the alpha motor neuronal pool of the antagonist. Hence, disinhibition. And finally, the 1A interneuron always causes excitation to the next neuron. This is false. I think it's repeated. So the answer is B, D, and H. B, D, and H. Okay, going on to the next question. Which of the following statements are true for a monosynaptic reflex? We have seen these reflexes, the H reflex and the M reflex in the previous class. Okay. Uh, so the H reflex or the Hoffman reflex is caused by an external stimulus. So the 1A afferents, because of the stimulus, the external stimulus in the nerve will contain both 1A afferents as well as alpha motor neurons. So the external stimulus, what it does is it causes a depolarization in all the neurons in the nerve, and the 1A afferents are the ones that reach threshold first for a lower amplitude stimulus, and then uh, travel along the 1A afferents through this synapse and then through the alpha motor neuron, and they cause an contraction in the muscle. So this is the H reflex, and if the stimulus amplitude continues to be increased, what happens is after a certain time, the amplitude is enough to cause the alpha motor neuronal, the membrane in the alpha motor neuron to reach threshold. So then directly there will be an action potential that is generated here, which travels and causes it causes a contraction in this muscle. So this is called the M response, which is directly due to the alpha neuron. So the latency of this M response is less because it does not have to travel through a synapse, whereas the latency of the Hertz reflex is more because it has to first travel through the 1A afferent, conduction through the 1A afferents, then it has to go through the synapse, travel through the synapse, the signal should travel through the synapse, then the action potential should travel through the alpha motor neuron. So that's the latency there'll be a latency that will be involved with the Hertz reflex, which is greater than the M response. So keeping this all this in mind, let's go back to this question. Which of the following statements are true for a monosynaptic reflex? Hertz reflex has a smaller latency compared to the M response. No, we just saw that the M response has a smaller latency. Both M response and Hertz reflex have the same latency. We have seen that the Hertz reflex has more latency because it has to conduct through the 1A afferent. The signal should pass through synapse and then through the alpha motor neuron. So this is wrong. The latency of the Hertz reflex decreases with increase in stimulus, whereas the latency of M response remains constant on increasing the stimulus. This is wrong. The latency, the path that the signal should travel to cause the Hertz reflex as well as the M response always remains the same. So the latency does not change for both these, the M response as well as the Hertz reflex. Latency of the M response decreases with increase in stimulus, whereas latency of the Hertz reflex remains constant on increase in the stimulus. Again, latency has no relation to the magnitude of the stimulus. The latency of both Hertz reflex and M response remains same upon increasing the stimulus strength. Yes, that's correct. Uh, The M response has a stimulus has a smaller latency because 1A afferent pathway is longer, including the M response has a smaller latency because 1A afferent pathway is longer, including a synapse where alpha motor neuronal pathway, whereas alpha motor neuronal pathway is smaller and does not include a synapse. Uh, Let us read all these three options and then we will see which is the correct one. The Hertz reflex has a smaller latency because the 1A afferent pathway is longer, including a synapse, whereas alpha motor neuronal pathway is smaller and does not include a synapse. This is wrong. M response has a smaller latency because the motor neuronal pathway is longer, including a synapse, whereas 1A afferent pathway is smaller and does not include a synapse. This is wrong. So this is the correct answer. The M response has a smaller latency because 1A afferent pathway is longer, including a synapse, whereas alpha motor neuron pathway is smaller and does not include a synapse. The answer is E and F. So whoever is watching the videos, they can first pause the video when the question is presented to them, and then they can attempt to answer the question on their own, following which they can listen to the explanation. So we are done with week 5 questions. We'll now move to week 6 questions, which the topics that are involved in week 6 that are there in week 6 are pre-programmed reactions, central pattern generators, spinal shock, uh, hyperreflexia, and all these topics. And we also start off with the motor cortex. So let's go through week 6 question. Let me just drink water. So has somebody joined in? Vikram Singh has joined in. Uh, Hello, Vikram.
Hello, good evening, sir.
Good evening. So I was just taking the live session. So I started. I started at 5:05, and I was discussing the week 5 questions, MCQ questions. Uh, so I'll continue with week 6 MCQ questions now. So the format is I'll first present the MCQ question, and then you can try and attempt to solve it. and then I will go in depth and answer all the discuss the question and the concepts related to that. So that is the format that I'm following. Uh, so I'll continue.
Sure. Thank you.
So the first so now we are done with week 5 questions. I'm going to start with week 6. So the first question is, arrange the following in increasing order of the recovery time after a spinal injury. These are the four options. A. Humans, monkeys, cats, and rats. B. Rats, cats, chimpanzees, and humans. C. Cats, rats, humans, and monkeys. Or D. Uh, Monkeys, humans, cats, and rats. Uh, so you can just attempt to answer this if you know. You can type out your answer in the chat box. Uh, it doesn't matter if it's wrong. Uh, we'll still go through the. You can still give your response, and then I'll give an explanation. Okay. So I'll just discuss this question now. So usually after spinal damage, uh, so there is a period where the reflexes are not present. So that is the situation where the person is going through a spinal shock, and then following that, the reflexes are back with the bang, and they are heightened. So that's a situation of hyperreflexia. And during this recovery time, so this hyperreflexia is there throughout the recovery time. So it has been noticed that as we go higher up in the species, the recovery time increases. So in this case, we have these four. We have uh, so the answer should be rats, cats,
Okay.
chimpanzee, and humans. So it takes. Uh, I'm able to hear my echo.
No, clear, sir.
Yeah, you can just mute. Yeah. And if you have any question, you can just type it in the chat box. Okay. So this is the situation. The answer is B. So usually in rats, the recovery happens within a few hours. In cats, it happens between an hour to 1 day maximum. Uh, in non-human primates like chimpanzees, uh, it happens with the recovery period is for days, so maximum a week. And in humans, it can go on. The recovery rate can be from uh, weeks to months. So why is this the case? This is because uh, the reflex as we go higher up in the species, the influence of the higher centers like from the brain and the cortex is more when compared to lower species like rats. So this is the explanation. The next question is, a condition where the reflexes are observed to be exaggerated is called as hypertonia, hypotonia, hyporeflexia, or D, hyperreflexia. So you can take around 15 seconds, 15 to 20 seconds, and whatever you remember, you can just respond in the chat box, and then I will give an explanation after that. hypertonia. Okay, so you have gone with hypertonia. Yeah, the okay. So I'll just discuss this situation. Uh, hypertonia is the condition where uh, the so if we measure the EMG, there is some baseline activity always in the EMG. If we take an electrode and we put it on a muscle belly and we record the EMG activity, there's always some baseline activity. So when this baseline activity is heightened uh, more than usual, then the situation is that is the tone, or there is always some tension in the muscle, uh, extra tension in the muscle. So that situation is hypertonia. And when the activity is very less, that is the tension in the muscle is lesser than usual, then the situation is hypotonia. And then we have hyporeflexia. So we have seen that we have seen reflexes. For example, we have seen the T reflex, the H reflex. Uh, so if the response of for these reflexes is depressed or less, then the situation is called hyporeflexia. And if the response of the reflexes is heightened beyond the normal response of the reflex, then it's called hyperreflexia. So a condition where the reflexes is the key word here. The reflexes are observed to be exaggerated is called as hyperreflexia, because like I just explained that the reflex response is exaggerated. So the answer is D. Going on to the next question. Uh, Spinal shock is a condition where the reflexes are exaggerated due to injury to the spinal cord. B. A period after spinal injury during which there is exaggerated reflexes. C. A period after spinal injury during which there is absence of reflex. Or D. A shock treatment given to the spinal cord to increase rate of recovery. So again, you can take around 30 seconds and you can just type out your answer and then we'll discuss. Okay, let's see. Okay, Vikram is saying A. Okay. So usually what happens is after a spinal shock injury, uh, there is a period where the reflexes are is not is absent, or is either very less or absent. So following which the reflexes during then following this there's uh, the recovery period starts, and during this recovery period the reflexes are very heightened. So this period where following spinal injury, when the reflexes are very less, that is called spinal shock. And when the reflexes are heightened, then it is called hyperreflexia. So it has been the exact reason is not known, but it has been hypothesized that the spinal shock can be due to decreased facilitatory inputs from the central nervous system or the higher centers. So that could be a reason. And why this happens, why hyperreflexia happens, is still not completely known. The exact reason is not known. So let us go through the options. A condition where the reflexes are exaggerated due to injury to the spinal cord. No, uh, this is hyperreflexia. A period after spinal injury during which there is exaggerated reflexes. No, a period after spinal cord injury where there is absence of reflex. Yes, this is the correct answer. And a shock treatment given to the spinal cord to increase the rate of recovery. This is wrong. The answer is C. So let's go to the next question. Concept of central pattern generators is well demonstrated in humans, and existence in lower animals is not known. Well demonstrated in lower animals and existence in humans is not known. Well demonstrated in both humans and lower animals. Not known properly in both humans and lower animals. So again, you can take 30 seconds to answer this. Okay, let's see what the response is. B. Okay, Vikram is saying B. Let's see. Yeah, that's actually correct, Vikram. So, uh, central pattern generators are mostly seen in lower animals, and they're not. It's still controversial. Their existence in humans is still. Uh, I mean, there's a lot of controversy whether the central pattern generators exist in humans or not. So it is well demonstrated in lower animals. So this is the correct answer. Answer is B. So let me just go through, open up that slide which has central pattern generators. You can just go through the slide to get an idea. So central pattern generators is a neuronal network within the central nervous system that is capable of generating a rhythmic pattern of motor activity without phasic sensory input from peripheral receptors. So this is seen in animals, for example, birds that are flapping continuously to fly, or it is seen in fishes that are swimming, or also in respiration. So it is proposed that this is due to this circuitry, the central pattern generator circuitry. Okay, let's go to the next question. Error correction and adaptation is associated with A. Basal ganglia, B. Primary motor cortex, C. Cerebellum, and D. Premotor cortex. So again, you can take some time, around 30 seconds, and give your response. Okay, let's see what the response is. Vikram is saying B. Okay, so let's discuss this. Uh, so usually what the basal ganglia does is it's involved in gating movements. It is proposed that it is involved in gating movements. So it suppresses spontaneous activity. That is what the basal ganglia does. And primary motor cortex is involved in controlling movements. So it gives certain commands to the muscles, and then the muscles it goes. These commands go through the spinal cord, and other mechanisms come into the picture, and then finally the command is executed at the level of the muscle by contraction of the muscle, which leads to movement when the muscles contract in a synchronized way. Then it leads to movement. So the primary motor cortex is involved in that. However, the command that is sent by the primary motor cortex uh, might not lead to the exact desired outcome. So we have feedback that is going, and there might be errors in our movements. For example, I'm hitting. For example, I'm learning how to play tennis, and I'm learning how to serve. So it's not that I might give a command to serve properly, but there might still be errors. So these errors are actually corrected by cerebellum. So there's error correction and adaptation that happens due to the cerebellum. So the answer is C. Okay. Now match the following. I'll give you around uh, 2 minutes to answer this. This is slightly a bigger question. Uh, I'll just set an alarm for 2 minutes. Okay. So you can take 2 minutes and then we'll discuss this. Okay. So let's see what the response is. Okay, Vikram has responded with C. So you have responded with C. Okay, let's discuss this. John Hewlings Jackson. So what John Hewlings Jackson actually did was, he said he was treating these epileptic patients, and he observed that so these epilepsy patients have seizures, and these seizures lead to movements. So what he observed was uh, the during the seizures the movements happen in move from one uh, part of the body to the other in a particular sequence. So it's not like random parts of the body are having these seizures, uh, and these movements somewhat mimicked natural movements. So what he proposed, based on his observations, so this is now known as a Jacksonian march. So what he proposed was that a region of the brain, of the cerebral cortex anterior to the central sulcus, has a causal role in movement. So that was his conclusion. So This was done by John Hewlings Jackson in the 19th century. Then we have Clinton Woolsey. So what Clinton Woolsey did was he did stimulation of a strip of neurons rostral to the central circus. So he tried to create a motor map in animals. He did this. So he tried to create a motor map of these animals using stimulation-based studies. So the strip of neuron rostral to the central circus, that is the same region. So and he extensively investigated this. So this is the correct answer. Then we have Sherrington, who so what he did was he studied the organization of the motor cortex in great apes, and he did this by using these microstimulation based studies. So that is Sherrington. And then we have the classic works of Penfield and Baudrillard. So so they did these studies in humans. Uh, so came up with the organization of the motor cortex in humans. Uh, so what they did was during surgery, way back in 19 in the early 20th century, uh, they were treating these people for epilepsy, humans for epilepsy, and they were doing surgeries. So during the surgery, what they did was, I think it was Penfield who stimulated certain regions of the motor cortex, that is the region rostral to the central sulcus, and what they observed was certain regions in the brain in the body were contracting, certain muscles in the body were contracting, and Baudrillard actually noted this down. So if a particular region in the brain was stimulated, then which part of the body responded to the stimulation by contraction. So Baudrillard was keeping a note of all this while Penfield was doing the stimulations. And what they found out was that uh, the regions of the hand, especially the fingers and the hand, uh, the proportion of the neurons that led to stimulation, led to contraction of the hand and the fingers was much larger than when compared to other regions. So other regions of the body. So there was a disproportionately large representation of the hands in the motor cortex, and this is one of the reasons why the hand is so dexterous. Uh, so they came up with this map, the homunculus, uh, which shows how many neurons, the proportion of the neurons corresponding to they corresponded this with the homunculus size. So the homunculus, the size of the hands was more because more number of neurons led to excitation of the hand. Similarly, the lip area, more number of neurons, stimulation of the neurons in the cortex led to uh, stimulation of the lip region. So the answer is 1C, sorry, 1B, 2C, 3D, and 4A. So 1B, 2C, 3D, and 4A. That's correct. You got the answer right, Vikram. So let's go on to the next question. A spinal preparation where the dorsal root ganglion is removed results in. So you can go through this and give your response. I'll just step out and get some water. In the meantime, you can go through the question and give your answer. Okay, Vikram has responded with C. Okay. So we have seen previously that the sensory information goes in through the dorsal root. So we have the 1A afferents, the 1B afferents, 2 afferents. So these and other afferents which carry this sensory information from all the receptors on our skin, the in our muscles, and this goes in through the dorsal side of the spinal cord. This enters into the dorsal side, whereas the motor commands, the motor neurons, which cause contraction in the muscles, which cause movement. So these are called efferents. So these neurons, the alpha motor neurons that cause movement and contraction in the muscle, goes out through the from the spinal cord through the ventral side of the spinal cord or the ventral root. So in the spinal preparation, what happens is the dorsal root ganglion is removed. So the dorsal root itself is cut. So this leads to a situation where there's no sensory input that is going into the spinal cord, and hence there's no sensory input that is being transmitted to the brain. So the answer is no command is passed on to the motor neurons. This is wrong. The motor neurons still have a root to cause movements, but no inputs from the afferent is being sensed. This is correct because the dorsal root has been cut out. Both afferents and efferents are disabled. This is not correct. Only afferent inputs are sensed. No, it is only efferent outputs that are given. So the answer is B. In immobilized preparations, L-dopa and nilamide is used to cause paralysis. The dorsal root ganglion is dissected to cause paralysis. Tuberculinine is used to cause paralysis. A spinal cord is removed and kept in saline bath and exposed to serotonin and NMDA to cause paralysis. So again, you can take around 30 seconds to answer this, and then we'll discuss this. Okay, let's see what the response is. Vikram is saying B. Okay, so uh, let us go through the slides that will give some more clarity. So in spinal preparation, what happens is the spinal cord is transected at the lower thoracic level, and this isolates the spinal segment that control the hind limb from the rest of the central nervous system. And then there is this acute spinal preparation where these drugs are used in the spinal preparation. But in this case, it's an immobilized preparation, not a spinal preparation. So this particular answer is wrong. L-dopa and nilamide. These are actually used in spinal preparation. Uh, so in immobilized preparation, what happens is the paralyzed in immobilized preparation, the muscles are paralyzed with deep tuberculosis, which is a competitive inhibitor of the acetylcholine that blocks synaptic transmission at the neuromuscular junction. So this is basically used to study the effect of proprioceptive reflexes. Uh, so the dorsal root ganglion is dissected to cause paralysis. No, that is not true. That is something that is there uh, in another type of preparation, not this. So this is wrong. we have seen tuberculosis is used to cause paralysis. This is correct. So the answer is C. And so if you recall from the initial weeks of lecture, what happens is this is a neuromuscular junction. Let us assume that this is a synapse, and then we have an action potential that travels here. So then we have voltage gated calcium channels which open up due to this action potential, and there's an influx of calcium, and we have these vesicles here which contain certain receptors that are sensitive to the change in the calcium level. So when the calcium level due to this action potential changes, these receptors which on the vesicles detect this, and they move this vesicle to the presynaptic membrane, and the vesicle fuses with the presynaptic membrane through these which is through these snare proteins, and they open up on the outside to release acetylcholine, ACH. Now this acetylcholine diffuses across the synapse, and it attaches to this uh, nicotinic cholinergic receptors. So when ACH attaches to these nicotinic cholinergic receptors, these nicotinic cholinergic receptors actually open up, and there's a huge influx of sodium that happens, which again causes a action potential in the postsynaptic membrane. So this is the basic working. Now what happens is tuberculine comes into the picture, and it competes with acetylcholine to attach to the nicotinic cholinergic receptors. So now if the tuberculine is attached to the nicotinic cholinergic receptors, ACH actually cannot attach to this, and hence it cannot open up the nicotinic cholinergic receptor. So this tuberculine basically blocks the nicotinic cholinergic receptors, and hence an action potential cannot be generated. So this leads to a situation where there's paralysis, there's no muscle contraction that happens, and because there's no action potential that is being generated, and this is how tubercularity works. So the answer is C. Which of the following is not related to pre-programmed reactions? A. Long latency reflexes. B. Functional stress reflex. C. M2 M3 response. Or D. T reflex. Again, you can take 30 seconds to answer this. Let's see what the response is. We, what is the response? Okay, Vikram has responded with B. Okay, okay. So for a pre-programmed reaction, uh, there are many names for a pre-programmed reaction. So one of those names is it's a long latency reflex. It's a functional stretch reflex. It's an M2 M3 response. Then there's also it's also called triggered reaction or triggered reflex, and some other names. So all these names refer to the pre-programmed reactions, and these names are given uh, based on the context in which the pre-programmed reaction is elicited. However, the T reflex is the tendon tap reflex. So let us remember the situation what happens here. So we have these receptors, the primary spindle endings in this is a muscle. We have a primary spindle ending, and then we have the 1A afferents. So this is the 1A afferent. So when we hit the with the tendon onto the muscle stretches, and then there's action potentials that travel along the 1A afferents, and then the 1A afferents actually excite the alpha motor neuron, which supplies this the same muscle. So this hitting the tendon The action potential travels along the 1A afferents through the synapse to the alpha motor neuron and then back to the muscle, causing a contraction. So this is a T reflex. So this is a monosynaptic reflex, and the response is stereotypical. It's mostly the same. So the answer is D, because it's a monosynaptic reflex, not a pre-programmed reaction. So this is the answer. Okay. So it's 5:58. We'll continue from this point onwards in the next class. So we have a few questions more to discuss. So we'll continue. We'll stop the class here. Uh,
Thank you, sir.
yeah. Thanks a lot, Vikram, for joining in, and see you next week.